Which of the following regions of an immunoglobulin molecule determines the idiotype? A. CH2 domain, B. FC region, C. Fab region, D. Light chain constant region, E. Hinge region. Correct. C. Fab region. Explanation: the Fab region contains the antigen binding site and includes the variable regions of the light and heavy chains. The idiotype is the unique antigen binding specificity of a B cell, and it's determined by these variable regions. Why not the others? A. CH2 domain, found in FC, involved in complement binding, not antigen recognition. B. FC region determines isotype, not idiotype. D. Light chain constant region, not involved in antigen specificity. E. Hinge region provides flexibility but doesn't affect idiotype. Question 2. A patient receives an antibody therapy that is unable to activate the classical complement pathway. Which structural region of the antibody is most likely altered? A. Fab region, B. Variable region, C. FC region, light chain variable region, E. Hinge region. Correct answer, C. FC region. Explanation: the FC region, specifically the CH2 domain, binds to C1q and initiates the classical complement pathway. Alteration here would impair complement activation. Why not the others? A. Fab region binds antigens not involved in complement. B. Variable region determines specificity, not complement function. D. Light chain variable region, same as above. E. Hinge region adds flexibility, not complement activation. Question 3. Which of the following functions is directly attributed to the FC region of an antibody? A. Neutralizing viruses. B. Binding to antigenic epitope. C. Determining idiotype. D. Binding to macrophage FC receptors. E. Undergoing somatic hypermutation. Correct answer, binding to macrophage FC receptors. Explanation: the FC region binds to Fc-gon receptors on immune cells like macrophages and NK cells, triggering processes like phagocytosis and ADCC, antibody-dependent cellular cytotoxicity. Why not the others? A. Neutralizing viruses, done by Fab region. B. Binding to epitope, Fab again. C. Idiotype, determined by Fab's variable region. E. Somatic hypermutation, occurs in germinal centers, unrelated to FC region directly. Question 4. The isotype of an immunoglobulin, e.g. IgM, IgG, IgA, is determined by which structural feature? A. Fab variable domain, B. Fab constant domain, CFC region, D. Somatic recombination, E. Light chain type. CFC region. Explanation: the FC region, specifically the constant region of the heavy chain, determines the antibody's isotype. Switching from IgM to IgG, for example, involves class switch recombination at the FC domain. Why not the others? A. Fab variable domain determines idiotype, not isotype. B. Fab constant domain, still part of antigen binding, not isotype defining. D. Somatic recombination creates diversity, not isotype. E. Light chain type. Light chains can be kappa or lambda, but don't affect isotype. Want a mnemonic?